Welcome, everyone. I hope you can all hear me. My name's Alasdair Douglas. My family is one of 6 riparian owners of the Western Ross Carron. Together we comprise the River Carron Conservation Association. We're here tonight to present and discuss the independent report of the Rivers and Lochs Institute of the Highlands and Islands University at Inverness, now known as UHI. And the Rivers and Lochs is now the Institute of Biodiversity and Freshwater Conservation. And we're presenting our talk on the analysis of DNA samples from caught and trapped fish and an examination of how stocked fish from a single year of broodstock crossings, that's in 2014, have contributed to the returning adult catch. Just a little bit of housekeeping, if I may. We put you all on mute so that the speakers are not interrupted. We'll have a Q&A session, question and answer session at the end, and it would be helpful if you put your questions on the chat box, and one of us will read them out as we get to them at the end of the talks. If we are running out of questions or someone has a burning question, you'll have to put your hand up physically just to attract my attention, and we can go from there. So let me just introduce the other people with us tonight. Delighted to introduce Melanie Smith from the Rivers and Lochs Institute. Melanie's a professor of biodiversity, conservation and education, and an assistant principal at UHI. We're also delighted to welcome Melanie's colleague, Dr. Bernd Hänfling, who's director of the Institute of Biodiversity and Freshwater Conservation, Rivers and Forests. Bernd is one of the leading scientists in the UK in the field of DNA-based biomonitoring. Bob Kindness needs little introduction to those who care about the survival of wild salmon in Scotland. Bob is the Carron's scientific and fieldwork expert. This evening we'll highlight just one chapter of the story of the River Carron, but the full story is really Bob's story as the guardian of the salmonid population for more than 25 years. Lastly, let me introduce Shaun Macdonald, who's owner of the New Kelso beat of the Carron. Shaun's a farmer and an experienced and expert salmon fisher. Shaun, together with the McPherson family from the Attadale beat, has supported the conservation work in the river since it first began, not only financially, which they've done in a big way, but more importantly, Shaun supports the work with a huge amount of enthusiasm for the project and his insistence on injecting the element of fun into every aspect of what we're trying to achieve. So this evening's intended to inform and generate discussion around the place of stocking in salmon conservation, and to ask you to support the further DNA analysis that the UHI wishes to do with our unique store of samples taken from caught and trapped fish. We think that the UHI studies are of global importance to those interested in the survival of wild Atlantic salmon, and allied to the science, our fieldwork and studies on stocking, we can give future generations of scientists and river owners the tools, we think, to bring back a river from the point of extinction. Shaun, can I hand over to you just to say a few words, and then we'll press on.
Thanks, Alasdair. Yes, I'm just thinking back to our trip to Edinburgh to the New Club, which was great fun and of great interest. And what I discussed very briefly down there, or touched on, was where we were and how we ended up where we are now. And I'm going back to the mid-80s when we still had a pretty healthy run of salmon and sea trout. At New Kelso, for instance, we averaged about 84 salmon. But we were in discussion with Marine Scotland. I think they were called the Freshwater Fisheries Division at that time, and Ross Gardner. And the conversation was basically, my late father corresponded with Mr. Gordon and Mr. Ewan McPherson, and then inviting Ross Gardner to give us some advice with the advent of fish farming in the area, which, while we had concerns, our other concern is we shouldn't stand post Shaun, post the oil yard being closed down, which employed 3,000 people, I think, at the time at Shaun, that economically fish farming was very important to the area. And Ross Gardner took the view that this was probably a sensible option. And in the correspondence, we discussed about restocking. Not at that stage for anything other than supplementing our what we had, or if anything should happen. Well, within 10 years, we'd had a collapse, and we were down to averaging 4 or 5 salmon in the system instead of averaging about 200, 250. What Ross Gardner had made perfectly clear is one thing we shouldn't be doing is taking any fish from anywhere else, which we had been doing in the past from the River Carron and on the Shin. And we'd stopped doing that, and he made it clear that we should take eggs from our own fish, genetically our own fish, and put them in at different times. It was wise words. When we actually did have the collapse, we weren't convinced it was entirely fish farming's fault. And we very quietly went about our business of trying to rebuild, which Bob will talk about, rebuild our stock and create a smolt run to challenge the fish farmers, basically, for safe passage. This is, as you've all read, been hugely successful. And it's all mainly down to Bob's perseverance and hard work and brilliant fish husbandry. What happened after that, and I'm trying to be as brief as I can here, is that we thought that people would start to recognize the work that Bob was doing, and how we had regenerated the river from a very low point. And you'll see this in the graphs. We went to a conference in 2011. It was a 2-day salmon conference. All the good and the great were there from Atlantic Salmon. And we were basically ignored. And the message was, you haven't got any science. You can't back up that the regeneration of the river is through the hatchery. And therefore, we're not going to take anything you say terribly seriously. And when we came back from that, and I remember it was very, very wet, we drove back up north, Bob and myself in my truck. And from that point, Bob started taking all the DNA, and took all the DNA from every fish that we bred from, and took the DNA from every fish that we caught subsequently, when all of a sudden, the success continued and we started to catch more and more fish. We didn't say much about it. And in between time, a lot of people have passed comment about what's been going on in the Carron. It's pretty simple stuff. And Bob will explain that to you about how he runs his hatchery. And Bernd will explain about the genetics. What we have now, thanks to the UHI and the perseverance of Professor Melanie and others, what we now have is the kind of concrete I put in when I've got a lorry going over the top of concrete, at 45 ton and turning on it. It's absolutely rock solid. The fact that the hatchery now not only is improving the catch, but may be a template for the future for other rivers when catches are very low to regenerate. And the question will come round, and I'm sure it will come round later, is when do you intervene to try and help a river that is close to extinction? There's other elements of that in terms of genetic integrity that we'll discuss later. The others are far better qualified than I am to do that. But I think we're at a stage now which is a fantastic place for us to get to, where we've read this initial DNA. It has given us confidence. It has, for Bob, it's vindicated all the fantastic hard work that he's done. And I would hope we will now have a sensible discussion about how DNA can be used as a fantastic tool, as it is in farming, as it is in a farming business, to enhance the salmon population and in turn enhance biodiversity in a river. So, Alasdair, at the moment, I have nothing else to add. I think
Thanks,
we should
Shaun.
just move on.
Yeah, that's super. Melanie, are you ready to come in now?
I am. Hopefully everybody can hear me. Hello everyone. So thanks Shaun, and thanks for the opportunity to just say a little bit about the conservation principles which underpin the project. So as we know, salmon are critically endangered, even extinct in some rivers, and as Shaun highlighted, that situation was what the Carron was facing a number of years ago. So when we put in, when we started the project, we wanted to make sure that it was underpinned by sound conservation principles. So Megan, if you could go to the next slide, please. Thank you. So these principles are a summary really of principles which are underpin many conservation projects, and they are firmly around putting in place a sound research and knowledge exchange. The knowledge exchange means working with all the partners concerned, ensuring that they have an understanding all the way along of what's happening, and that there's good communication along the way with all of those partners that are involved and the communities. Establishing a baseline, and when you hear about the genetics work that Bernd's going to be presenting, that genetics work is very much showing the way in which genetics can be used to put in place a baseline against which you can then monitor the rest of the recovery, hopefully, of the species that you're looking at. In this case, obviously, salmon in the River Carron. Establishing that program of monitoring, and that was what Shaun was alluding to, a rigorous program of sampling underpinned by protocols, which mean that when the fish are handled, it's all done in a carefully controlled way. And then ensuring there's this cycle of review and reporting, and that communication and engagement piece, which is very important. Obviously, in UHI Inverness, we've been very concerned with making sure that we've been fully working with the partners, and I have to say I have had the pleasure of being able to do that, and the honour of being able to work with all of the River Carron Conservation Association, and it has been a really good partnership as we've worked together, because we've needed to remain independent doing the science work, but also develop and communicate what we're doing with the science. So I'm not, that's really my piece here, and this is just to introduce and set that scene. It is now nearly 20 years that I've been involved with the project. Most recently, Bernd has come on board to lead the genetics side of the work with our colleague Victoria Pritchard, but I've worked most closely with Bob over the years and with Shaun, and I'm now going to hand over to Bob, who will tell you about his work that he's done over the years following the collapse of the Carron. Over to you, Bob.
Thanks, Melanie. Yeah, the Carron really had a major collapse towards the end of the 90s. It's a case of what caused this collapse, and it seems to be that a series of big winter spates, I think, would have washed out most of the young stock in the river. And you can see in the figures here that in 1988-89, there was a huge spate that year, and I'm pretty sure everything would have gone. The river was completely in flood. Huge areas of banks disappeared. Thousands of tons of gravel went down the river. It really was a mess. That would have been survivable had it been a one-off 1 year. But unfortunately, over the next 4 years, we had major spates every year. And that would have meant that by the end of that period, virtually everything would have gone from the river, and there would have been nothing at sea to come back in. So that would have caused a very rapid collapse. I've included there 98, 99, and that year James Butler, who was the biologist for the Westeros Fisheries Trust, he did a project looking at most of the rivers in Westeros to see whether red washout was an issue. he did 5 sites in the Carron, and in every one of those sites, he found that there would have been washout. He used beads buried in the gravel to see whether that would have been equivalent of eggs, whether they had been washed out. Now, the biggest spate that year was only 120 cubic meters per second of water flow. And if you compare that with those 5 years, it was a lot less. So there's a fair chance, and I'm pretty sure definitely most of the stuff would have been washed out in those early years. I've included 2006-07. In that year, we had a spate at 233 cubic meters per second, and we found very little during electrofishing the following year in terms of young fish. So it's fairly clear to me that would have been a major contributing factor to the decline. The problem then with losing stock, you can see from the cycle there that you'll finish up getting poor egg deposition, resulting in poor numbers of juveniles, low smolt output, lack of returning adults, so the cycle goes on. And it'd be very difficult to see how that would change. You can see in the graph on the right there how the numbers dropped, and they dropped very quickly from quite reasonable returns through the 80s, as Shaun alluded to virtually nothing as we went through into the 90s. Next slide. Right, so it was a case of what would we do about it? Now, there was very little in the river to use as broodstock, and the problem with a stocking program is you need quite substantial numbers of eggs to actually make a difference. So we had to go down the route of establishing our own broodstock, and that was done by taking what we could get from the river, get the eggs from those hen fish, and take a proportion of those eggs right through to become broodstock, and that was done entirely in fresh water. And you can see the results there in the graph. The numbers recovered quite dramatically. It wasn't a case of a very slow recovery. They recovered very quickly once the stocking program got going. The numbers of fish we put in over the years, the actual total up to now, up to last year, was 4.6 million at different stages have gone in. And the biggest number went in as fed fry. We found that over time that these seem to be giving us the best results. So there was over 3.5 million fed fry have gone in over the years. Next slide, please. The hatchery we use is very simple. I operate on 3 different sites. No site has any buildings. We're all working, they're all working outside. I don't use any electricity, and it's a very simple system, a very simple throughput water system. Fish don't need buildings. Fish don't need anything fancy. They just simply need a really good water supply and someone that knows what they're doing. And we were able to achieve that. Now on that slide, I've been given an indication there of how successful fish are in the hatchery, how successful using broodstock is. These 6 fish, now it's quite interesting because these 6 fish caught in 2019, they actually had come from the stocking program. I had taken them from the river to use as wild broodstock, and unbeknown to me, until the DNA was done, these were actually fish that had come from the stocking program. And the results were incredibly good. As you can see there, 99.3% of the eggs came through to the eyed stage. And I know from many years' experience, if the eying rate is that high, then the stock is particularly good, and they will be good all the way through the cycle. One of those fish was a 22-pound hen that came from the Attadale broodstock, the captive broodstock. She produced 11,450 eggs, and by the time those fish got to 2 to 3 grams in size, I still had 11,200. So that gives an indication of how successful these fish were. And the importance of that is that these fish are then in your system, these stock fish are then back in your system, and they will breed and they will produce successful offspring. Next slide. The argument would be, you know, how can you prove that the stocking is due to, the recovery is due to stocking? That's something that might be due to something else. If we look at this graph, in the early days when we started, without the captive broodstock, we were dealing with relatively small numbers of fish going in. And as you can see from that chart, there wasn't really a huge increase in catches. Once we got to the offspring of the captive broodstock going into the river, and that was 159,000 in 2001, following those fish through to go to sea as S2s and come back as grills, then the catches shot up to 140 in 2004. So it was quite clear that something quite dramatic had happened. The next few years after that, we would still have been relying very much on stockfish to give us our continual increasing in catches, increased catches. It wouldn't have been until 2008 before natural stocking would have contributed greatly to the catches. It's important, obviously, to monitor the stocks, to know what's happening, to get an indication of whether the stocking is working or not. And to do that, we had to set in place various projects. One was a tagging project that we did over 3 years in 2006, 2007, and 2008, where we put CWT tags into 105,000 autumn salmon, and these were fry of around 4 grams in weight. And then we were able to monitor these fish using a rotary screw trap. These fish were all, as well as being tagged, they were also fin clipped, so we could identify them without having to kill the fish to check for tags. We found through the screw trap that quite substantial numbers of these fish did leave the river as smolts. In 2010, 822 clipped smolts left the river. Some had also left the year before as small S1s, but a very small number had left as S3s the next year. And it actually represented a very good proportion of the stock fish going out of the river. Next slide. Monitoring is key. I'll pass on to Alasdair now to cover this a little bit on the details of the DNA project.
Yes, I'm just going to say very quickly that monitoring is the key aspect that we focus on. Conservation generally requires that very close monitoring to see how things progress. Bob has collected a huge number of samples from the fish over the years. We've got some 6,000 samples of DNA, little clips off the tail of the caught fish, and we've had UHI analyze just over 800 samples so far, and it's the 800 samples that we're talking about tonight. What we want to do, and I'll just talk a little bit about it at the end, is to raise a bit of cash to do the sampling of the remaining 6,000, which will give us a much clearer and much more robust set of figures to do our analysis on. As I say, we'll come back to the amount of money we've spent so far and what we intend to spend going forward. So back to the next.
The broodstock that we've used over the years, there have been 3 different broodstocks that contributed to the 2014 eggs. Wild broodstock taken directly from the river. Obviously, once the catches started to improve, that became a possibility again, which wasn't really there in the early days. The Attadale broodstock, which is the one of the captive broodstocks, that was the first captive broodstock, and these fish were reared entirely in fresh water. The eggs were hatched, the young fish taken through, and then the stock was taken right through to become fully mature. The third broodstock is one that came in later, and this was one that we were working with the local fish farming company, Scottish Salmon Company. They were keen to look at using Scottish stocks as opposed to simply using Norwegian stock. So we provided eggs for them that came from multi-sea winter hens crossed with multi-sea winter cocks. And these fish were then reared through in fresh water to the smolt stage and then in sea cages to become mature. And this was all done out in North Uist. But these were all native Carron stock. Next slide. This is an example of a stockfish. This was one of the clipped fish from earlier in the program. And it demonstrates that a stockfish is every bit as nice looking as a wild fish. I've got quite a lot of stockfish over the years. Some I recognize as stockfish and some I couldn't tell from wild fish. In fact, one of the ones that was caught in this DNA study that we've just done, one was a 23-pound cock. And when I caught him, I must admit, he was one of the best looking fish I'd seen. I had no idea that he was a stockfish. He simply looked like a perfect wild fish. So it's a bit of a misconception that stockfish are somehow inferior in terms of appearance. And I would defy any angler to tell them apart from straightforward wild fish. Next slide. The hope is with stocking that you'll make a difference, but you have to remember with stocking it is a numbers game. To be successful, you have to be putting decent numbers back into the river. And in reality, you're trying to emulate what might happen in the wild, but obviously with the hatchery, you have a much higher success in terms of survival. The hope is that by increasing juvenile numbers, you'll increase your smolt run, which is definitely what we found, and we were able to prove that using the screw trap, that a much higher number of smolts are leaving the river than in the past. And if you look at that little chart there, it gives an idea of the sort of numbers of fry you would need for every rod caught fish. And there you're looking at least 2,000, 3,000 fry will produce one fish to the rod, but that will also produce another 5 or 6 fish coming back to the river as brood fish that hopefully will spawn naturally in the river. Next slide. I'll now pass on to Bernd.
Oh, good evening, everybody. First of all, thank you very much for the River Carron Conservation Association for inviting me. So I've been asked to talk about the contribution genetic approaches can make to the conservation of the River Carron salmon population and maybe more widely to salmon conservation. I've only been appointed as the research director at UHI since March and haven't really had a chance to meet many people in the room or from the Scottish salmon world more broadly. So I thought I'd start off introducing myself to give you a little background about myself, and hopefully it will be a bit less strange than why this bloke from Germany talks about Scottish salmon. Next slide, please, Megan. So here are some pictures from my quite personal album with an added comedy value of some 1980s haircuts. I'm actually getting slightly worried now since I realized this is going to be recorded and these pictures are going to be online for everybody to see. But I've been told that these haircuts are trendy again now, so maybe it's not a bad thing. So I grew up in the Bavarian countryside, and really from an early age, I spent most of my time out on local trout streams fishing. And as a teenager, I got the amazing opportunity to manage my own trout stream. It's only a small stream, but it had fantastic fishing, like this 3-pound trout you can see on this picture. And maybe what is relevant in the context of what we are talking about today is actually at the time in Bavaria, there was an obligation for fishery managers to meet annual stocking targets. For example, I actually had to stock, I was legally obliged to stock with a certain number of 0 to 2+ fish every year. So during my undergraduate degree, I then started volunteering for the Bavarian fisheries authorities, participated in every activity I could. This included hundreds of hours of electrofishing to contribute to the first distribution atlas and work in the model hatchery, monitor fish passes, everything really I could do relating to fish and fisheries. Next slide, please. So when I moved on to do a PhD project, I did this in collaboration with the fisheries authorities, and I looked specifically at the genetic differentiation of fish populations across different river catchments. Now, this was something what nobody really knew about at the time. Nowadays, we are very familiar with the fact that fish from different rivers are genetically quite different, but at the time in the 1990s, this was really not very well known. So what I found really that fish from different catchment, almost from every river, were genetically very distinct. And together with other studies which were carried out at the same time, this really contributed to a rethinking of stocking, in the sense that people started to consider that we need to only stock with local fish rather than transferring fish across different catchments. So this project made me realize what an incredible tool genetic methods are to obtain a huge amount of information for conservation and fisheries management. So I did continue to apply these tools to freshwater science for 25 years. And most recently, I've led the development of environmental DNA-based monitors to approaches to monitor fish communities. So what we can do now is we can just take a few water samples from a loch or from a river, and we can reconstruct the fish species composition from these samples. Next slide, please. So in my job at UHI Inverness, I'm heading the newly formed Institute for Biodiversity and Freshwater Conservation, which was formed by merging what was the Rivers and Lochs Institute, so the institute which carried out the study we're talking about today, and the Forestry and Conservation Group. By moving these two groups together, this allows us better to exploit the synergies between these different areas and exploit new opportunities. And most importantly, it allows us to work on a landscape and catchment scale. There has been a really rapidly increasing awareness that as part of conservation strategies, we need to consider what is going on across the wider ecosystem. For example, to conserve salmon populations, we can't just focus on the river environment, but we need to understand what is going on the land around the catchment. How is the land used? What is the tree planting around it? Because this will have an impact on processes within the riverine environment. Next slide, please. So sorry about the slightly extended introduction. I hope this was of interest in this context. But what I want to do now is I want to talk about a specific project the team at our institute has carried out using genetic methods to evaluate the Carron stocking program Bob was just talking about. I have to emphasize that I wasn't involved in this project at all. This was before I was appointed, and I'm not a salmon specialist either. I've been asked to present the project, which was led by Victoria Pritchard, which unfortunately was unable to make it today. So the genetic approach we employed in this project was very similar to what is used in forensic science and, for example, in genetic paternity testing. Apart from the fact that what we are doing, we're actually using a much deeper scan of the genome. So we actually have better data than you would generate in these kind of approaches. But basically, we are creating genetic fingerprints of individuals from fin clips, which are taken from hatchery and wild fish. And this allows us to address a number of really important questions. So first of all, it allows us to identify whether a returning fish originates from a hatchery or from wild spawning. And this is obviously very important as it shows whether the stocking program is actually effective, whether it actually works. So as we get information from a large number of individuals from that population, we can also use this to assess the genetic diversity of the population. So are these fish quite similar to each other, or are they quite different? And this genetic diversity is really important because it provides the raw material for a population to respond to new disease challenges or changing environmental conditions, such as rising temperatures or different nutrient contents and so on. And what we know is that populations with a low genetic diversity are much more likely to become extinct. Now, that is really very well established in the literature from theory and also from lots of empirical studies, not only on salmon. So processes which lead to low genetic diversity are small population size, and inbreeding, and we know that these two factors are bad. So the third issue we looked at in this project is the genomic contributions from salmon which have escaped fish farms, and that is unfortunately quite a common problem. And this is an issue because these aquaculture fish were obviously selected for traits which are useful in aquaculture conditions, but they're less useful in the wild. So if these genes get integrated into our natural populations, they can decrease the fitness of wild populations. So next slide, please. Oh, I think I skipped one slide. That should have been the background from the last one. Yeah, the genetic tagging or genetic parentage analysis works. So one more. So the genetic tagging I've just talking about, or the same as parentage analysis, works on the principle that we know that the offspring inherits exactly 50% of the genome from each of the parents. So we can predict exactly what the genotypes of the offspring from each breeding pair would be and what genotypes they would have. And we can check each individual fish which we catch in the wild and whether their genotype match to a predicted genotype from parents in the hatchery or not. So the stocked fish will have a genetic match with a broodstock parent, the wild fish will not. Next slide, please. So in this project, we were able to genotype the broodstock from 2014, just from a single year, and following the small generations from 2016 and 17. So these are the cohorts which we expect from that year to become smalls, and the adults we expected to return between 2017 and 19, but largely in 2018. So there's a bit of a variation of when the salmon exactly return. Next slide, please. So this is really the key figure which shows the results from these genetic assignment tests. This pie chart shows the relative contribution of the 2014 broodstock in red. So you can see this for the 2016 and 17 smalls, and for the 2018 returning adults. So in these cohorts, about a third of the fish were from hatchery origin. But this is really good news because it shows us three important things. First, hatchery fish are clearly surviving this smolt stage in good numbers. Secondly, they are returning in good numbers. And finally, the survival of wild and hatchery smolts hatchery fish seem to be very similar, because the proportion of hatchery fish in the smolt and returning adult cohorts are quite similar. That means these two different cohorts must survive at a similar rate. What these figures don't show very clearly is, of course, there are also in the 2018 returning adults, for example, there will be other hatchery fish from 2013 and 2015, which we can't actually trace back because we haven't genotyped the broodstock from these cohorts. So in fact, these figures will be underestimating the amount of returning fish. And we estimate that the real numbers are in anything between about 35 and 50% of the fish will be actually returning fish from the hatchery. So what we really need is to really get a better understanding of this. We need urgently data from other year to complete this this picture. We need to genotype the broodstock from all the other different years, first to see what is the total number of returning adults, but also whether this is really consistent. Was this a one-off, or do the fish really consistently return back to the river from the hatchery? The next slide, please. So now to the issue of genetic diversity. This graph shows the genetic diversity of the different broodstocks here in green, orange, and blue, and also as a baseline the population for the population diversity of other wild-caught fish in gray. So now I have to emphasize that the differences you see between the different cohorts are quite small. I actually have to zoom in onto the graph to make these visible. But there are differences. So we can see that the broodstocks do have a lower genetic diversity as the wild-caught fish. And what you can also see that within each of these broodstock groups, the parents have a higher diversity as the offsprings, and that is quite consistent across the three different cohorts. Now we don't really know whether this is a random variation due to sampling, because we only have data from this 1 year, so we urgently need to get more information from more generations to understand whether this is a consistent trend caused by the small population size. Because what really matters when we're talking about loss of genetic diversity is a trend over time. And if this is a consistent trend, then this is something we need to worry about. Next slide, please. So the reason why we need to get accurate information about the trend in genetic diversity is that we need to make sure that we're not losing genetic diversity at a damaging level. And I mentioned this before, how important genetic diversity is. What we know very well from theory and many empirical population studies is that population diversity decreases over time, as this theoretical model here shows. And this is true for any real population, and it depends on population size. However, if the loss is small, it can be compensated through new mutations and immigration. But if it is significant, then the population diversity will get into a downward spiral. So what we need to know basically is, where are we with the salmon population? Here, are we in the blue area where we don't need to worry about the continuous loss, or are we in the red area? In which case we need to do something about increasing family size, for example, family numbers, and maybe do more targeted crossings to generate offspring with a maximum genetic diversity. The last question we addressed is whether the River Carron population has genetic ancestry from Norwegian aquaculture fish. Now, these genetic plots are not very easy to read, but essentially these blocks consist of a lot of different vertical lines. Each line represents the genetic structure of an individual and shows the proportion of native gene pool and aquaculture origin. So if a line is entirely blue, the fish has got pure genetic origin, and if there's any red or orange contribution, it represents a proportion of aquaculture genes. The take-home message is there is a small but not negligible amount of aquaculture genes in all of these broodstocks, but also in the wild-caught fish. From the current data, we don't really know what is the contribution of the captive rearing process in maintaining these genes. So we need a longer-term data time series to analyze that. Next slide, please. So I've mentioned a number of times that we need a larger data set. We look at these trends over time, looking at broodstocks from genotyping broodstock from many, many years, and also the returning fish again. And here we actually have the unique opportunity to have a tissue bank, which Bob has collected over the last 15 years, from fish, from broodstock, and from returning fish and from smolts, which we can readily use for such an analysis. Next slide, please. So utilizing that and genotyping these remaining tissue samples will allow us to provide really solid evidence to the question which I've outlined in this talk, but also to additional questions. For example, what is a relative reproductive success of stocked versus wild spawned adults? So when these stocked fish come back, is their reproductive success comparable to fish of a completely wild origin? So ultimately, this will provide solid evidence of what the role of supplementary stocking programs in the quest of protecting this magnificent species can play. And so I think we have an opportunity here to establish an experimental system which can be used to used to investigate other aspects of salmon conservation, for example, including the impact of man on the environment on a catchment scale. So the existing data and the knowledge and the tissue collection is really quite exceptional. And although I'm not a salmon specialist, I'm not aware of anything comparable in the UK and possibly not in Europe either. So it is really quite a unique opportunity to provide, as Alasdair said, research which matters on a worldwide scale. Right. So with that, I have finished and pass on to the next person.
Bob, are you taking this, or should I say a few words? I think I was going to just echo what Bernd was saying about the opportunity here and give UHI a bit of praise. It's got unbelievable facilities, and this is groundbreaking research for them and for the salmon community. And I think if we can keep going with this, then UHI can perhaps lead the way for the salmon community in keeping the Atlantic salmon from extinction, perhaps in many rivers.